Okay, good morning, everyone. Let's get started. So today we're going to be talking about how science is done, how it's disseminated, how you can evaluate good science, what makes science good, what makes science bad, and we'll talk about a few sort of cases of some really good science, and then we'll talk about some cases of really bad science, and then we'll touch a little bit upon the role science plays in conspiracy theories and stuff like that. So if you haven't already, make sure you do turn in your short paper. It was due yesterday at midnight. For some reason, Blackboard switched the times on me. So it was originally set to be due yesterday at midnight. I realized it was not actually midnight. It was like 6 p.m. and then I switched it, and then it switched to like 11 p.m. And I don't really know what happened, but just to let you know, if if it said it was late, but it was before midnight last night, it's still considered on time. I'll check the timestamps to make sure.
Professor?
Yep.
I have a question about
Chip. Sorry, hold on a sec.
That's okay.
Okay, one more time.
I had sent you an email. I don't know if you got it, but for the paper submission, it uses Turnitin, and it said to remove any personal information, and I wasn't sure what that meant, so I just removed like my name and your name and the biology class, like the heading basically from my paper.
Oh yeah, I did see your email. I wouldn't really worry about that.
I just wasn't sure if I was or wasn't supposed to do that because I didn't know what else qualified as personal information in the paper. So
yeah, I mean it tags with your name and your your UMass ID and stuff like that.
Okay.
So I know who it is even if you removed your your thing. But yeah, I mean I think they just do it because they don't want you to put personal information, because the way Turnitin works, it has like a bank of papers every time. Someone submits a paper, it gets sort of like stuffed into the bank in addition to the sort of the whole internet that it checks. So it doesn't want, I think it just doesn't want your personal information in that sort of bank.
Okay. All right. Thank you.
Yeah, no problem. So, yeah, so Turnitin was giving some people some problems. If you had problems with Turnitin, let me know. There's all sorts of weird issues with Turnitin, but yeah, so as always, if you have any questions, you can blurt it out or drop it in the chat. But let's talk about how science is done. But I actually want to talk about something a little bit cool. At least I think it's cool. You might not think it's that cool, but I think it's cool because I'm a microbiologist. So I'd like to talk about the blob first. And it's not going to be this blob from the Steve McQueen movie back in the 60s. It's going to be this blob. And this blob is a slime mold. And so many of you may have seen this last year. It was late last year. You would have seen this article like between October and December. But it's a cool like sort of story about life and how this sort of very simple life that could do some really complex things. So I thought it'd be a cool sort of time to sort of share this sort of funky guy and show you sort of how life has evolved these really interesting capacities, even though this particular form of life is very small and very simple. So the blob itself is a slime mold called Physarum polycepharum. I believe that's how you say the last thing. I'm terrible with Latin names, but it's what we call a slime mold. So it's not a prokaryote because it has nuclei. It has many, many, many nuclei. In fact, some of these slime molds can have hundreds of nuclei, as opposed to just our cells, which just has one nuclei. It's not a plant, so it's not photosynthetic, meaning it's not producing its own energy. And sort of the interesting things, it shares many traits with fungus. So it's just sort of in this nondescript category. But this is what the slime mold looks like growing in nature. They're very, very rare, sort of in urban environments. Like even if you went out to say like the Blue Hills or like Mount Wachusett in Western Mass, or actually that's Central Mass, but they're very rare in sort of these more urbanized areas because they're pretty sensitive to pollution. But if you go out to some really pristine areas of say the Berkshires in Western Mass, you'll start to see some of these oh so fun slime molds hanging around, eating dead wood and things like that. So slime molds themselves, again, they're not fungi, they're not plants, they're not animals. They're these sort of oddballs of the sort of world. They're unicellular. And so when we look at this picture back here, this is all just one giant cell, just one giant elongated cell. So it is typically a unicellular thing. There is the capacity for sort of slime molds to fuse together. So you can have two distinct slime molds become one. And so they sort of have this, this clustering ability. So it's kind of interesting. Like fungi and many plants, they reproduce with spores. And the thing that slime molds are best known for is chewing through dead wood. That's sort of what they do. But unlike our fungi and unlike many organisms, they are motile. Inside their cell walls, they lack this chemical called chitin, and that's sort of characteristic of all fungi. And the other thing that's kind of cool that they do is they hunt and digest their prey. And so they will literally, you know, just sort of inch along the ground hunting things to try to capture and kill them. So I think that is a sort of a wacky and wild thing that they do. And then finally, they're pretty common. There's about 900 species that have been described. We think there's probably at least another 10,000 more out there, but they're relatively common. But again, they are pretty sensitive to pollution. So the question you should ask yourself is, why am I spending time talking about this? Why should I care? Why did NPR pick on it? Why did all these news outlets pick up on it? And why is one on display at the Paris Zoo? And why is it getting all this stuff? Why is it getting all this fanfare? And the reality is it does some pretty crazy things. So the first crazy thing that they can do is they can repair themselves. So if you cut a Physarum in half, or any slime mold in half, it will essentially rejoin itself. Now, this would be the functional equivalent of me cutting my arm off, and my arm, not me grabbing my arm and sewing it back on or sticking it back on with glue. This would be like my arm crawling back to me and attaching itself back to me. So that's what these guys do. So they can reattach themselves pretty easy. They can also learn, so they can solve mazes. So as you can see in this image up here, this Physarum has solved this maze to get a tasty treat, and for a Physarum, its tasty treat is like an oat. So they can solve mazes. So that's a sort of an important thing. And you can sort of see how it's learning, right? It's sort of going through the maze. It encounters a dead end. It sort of breaks off and it traces its way back. And so it has the capacity to, at least rudimentary speaking, think and solve puzzles. Again, but it's just one cell. It's a single cellular organism, but it has the capacity to solve puzzles. Kind of a cool thing. If it's faced with an obstacle that it doesn't like, it will essentially learn how to get around it. So for instance, if I have this Physarum growing on a petri dish, it's where it's growing right now, and I put a drop of caffeine in front of it. Well, caffeine is toxic. Humans have sort of repurposed it for, enhanced performance at varying levels of our lives. But we have this caffeine. It's supposed to be toxic. We use it for other things, but caffeine would be toxic to Physarum. So if you put a droplet of caffeine in front of a Physarum or any slime mold, what it will do, it will sense the toxicity and then find a way around it. And the cool thing about it is if you present the Physarum or any slime mold with the exact same drop of caffeine or something with caffeine in it, it will instinctively know, without having to do any real thinking or any sort of real-time stuff, it will just know to inherently go around it. So it can learn things and it can remember things, which is pretty sort of extraordinary when you think that, again, this is just a single cellular. Mold. So the idea being is if we have a Physarum that learned that caffeine is bad, it fuses with another slime mold that didn't know caffeine was bad. Well, now the whole mega organism now knows caffeine is bad. And it's really sort of interesting. And the other thing about it is, like, doesn't matter how many slime molds you fuse together, they'll still act as one giant organism, and they'll all share their collective knowledge with one another. So again, it would be like, you took a physicist and a biologist and a chemist and different sexes of slime molds, and their sex is determined by their chromosomal arrangement and the genes they have on their chromosomes. So it's not determined by like XY, it's determined by the presence and absence of sort of specific genes as a whole. So, and this is what they look like when they're trying to, you know, reproduce. This is them getting ready to produce some spores. So, and sort of the thing I want to talk briefly about before we sort of move on to the topic of the day is their capacity to solve problems and how they are uniquely positioned to solve some serious problem. And so there's this really fascinating experiment that came out a few years ago. It's called the Tokyo Railway Experiment, or the Japan Railway Experiment. And what these researchers in Japan did is they took their slime mold and they stuck it in the middle, and they put a bunch of oats on a nice sort of nutrient medium for this Physarum to grow. And now what this corresponds to, if you know anything about the Tokyo metropolitan area, this is the Tokyo metropolitan area with the geography, as well as all the different major ports or the major stations within the Tokyo metropolitan area. And so what they did is they put the Physarum right in the middle, and they put a piece of food on all of the distinct major places inside the Tokyo metropolitan area. With the idea being is, how does this Physarum best solve this puzzle? How does it best connect these things? Because if you think about Japan, it has a very rich and very efficient railway system. And to develop that railway system, it took engineers years and years and years of planning and math to figure this out. And so the researchers asked, can you get this Physarum to figure out this really complex problem? And the answer to that is actually yes. And so you can look at how it sort of connects all these different pieces of oats together by its growing. And the idea being it's maximizing efficiency. So it's trying to get from oat to oat to oat by maximizing efficiency and connectivity, which is the exact same way you would want a railway line to do. You want to max efficiency and connectivity because that requires resources, that requires overall energy, the same way this Physarum does. And the cool thing is when you look at how the Physarum sort of orients itself and you look at an actual railway map, it's actually remarkably similar. So if you look at the slime molds map relative, which is here in C, versus the actual Tokyo Railway map, it is surprisingly similar. And it's similar enough that it's not sort of due to random chance, which I think is sort of an amazing thing, that this fungus figured out this really complex problem of how to connect all these different spaces with the least amount of energy, with the least amount of sort of waste, which I think is a really remarkable thing. And because if you think about it, right, this railway system here in D that we developed, well, that took years and millions of dollars and really, really, really smart people. So this is our slime mold. Again, it's a single cellular organism. So this giant thing on the plate is one organism. It's essentially just a giant bag with hundreds of nuclei in it. It has no neurons. It has no tissues. It is one giant cell. And so it does all this problem solving. It does this learning. It does this crazy reproduction, all with just being a big bag of nuclei. And so I think this is a really fascinating case to sort of see like life, you know, can be really, really complex even at the sort of single cellular level. And I think, you know, we like to sort of get caught up in this idea of bigger is better or bigger is flashier or bigger is smarter. But this slime mold, again, it really shows that you can do a lot of things at the single cellular level. So anyways, I just wanted to share that little anecdote with you. I'm pretty sure most of you, if you saw the blob or this thing, you would have glossed over it. But I think it's a really sort of funky, crazy sci-fi thing that I really enjoy sharing with people. But with that out of the way, we're going to sort of switch gears now. We're going to talk about science, how science is disseminated, and the topic of the day, the slime mold was just a distraction. So before we move on, we're going to just talk about some terminology first. And so the first sort of piece, we sort of have a couple different things we need to lay the groundwork for before we can sort of talk about these things. The first is a hypothesis, which is simply just a well-framed question. I hypothesize that plants will grow better with higher fertilizer because fertilizer increases growth efficiency. So that's a hypothesis. A theory is a well-supported hypothesis and is based on copious amounts of data. So the theory we spent the most time talking about this semester is evolution. As you sort of seen, there's tons and tons and tons of evolutionary theory evidence out there. And there's way more than I haven't showed you. But again, a theory in a scientific perspective is a very well-supported hypothesis. This is sort of in contrast to, average everyday person speak. And when I say, oh, I theorize this, well, it's me just sort of thinking out loud, right? Whereas opposed to in science, when you see something as a theory, it's something that has a lot of meaning. It has a lot of data behind it, a lot of evidence behind it. So a theory is kind of a big deal in science. Not everything makes it to a theory, but things do progress from a hypothesis to a theory as they gain more and more evidence. Now evolution started off as a hypothesis, and as it gained traction, it became a theory. Sort of the next rung up on the ladder is a law, and this is a theory that is essentially always true. And the theory that law that we talked about in this class was actually the law of thermodynamics, how energy cannot be created or destroyed. It can only be changed forms, right? When we talked about energy in class 2 or 3. So that's a law, something that is always true. So you think about this like gravity is a law, and gravity is always physically true, right? You throw a ball up, it comes down 100% of the time. You jump up in the air, you come down 100% of the time. And gravity is, the cool thing about a law is it's pretty consistent across the universe as far as we can tell. So gravity behaves the same way here as it does on the moon, as it does on Venus, and so on and so forth. So a law is something that's pretty consistent across the cosmos as well. An experiment is science that's done either in the lab, computer, or in nature. This is basically the idea of how you can test a hypothesis. If I have a well-framed question, my hypothesis, I can test it by doing a set of experiments. And science has really evolved from sort of this, I just study nature, to I just work in the lab, to now I can do all sorts of crazy things on a computer, like simulate things, run models, analyze genomes, things like that. So experiments are wide-ranging, and they usually combine more than one category. So if you do a lot of lab work, you're usually doing a lot of computer work as well. And then finally, we have what's called peer review. So experts in a field critique a submitted scientific article, determine its rigor. So they're looking at their methods, the statistics, sample sizes, the experimental design, all sorts of things to make sure that if this paper is put forth to the public, it's put forth to other researchers, that it is actually something that's scientifically sound. So when we talk about all these cases of evolution and all the things that we talked about in this class thus far, these were peer-reviewed by scientists that are very knowledgeable in what they're reviewing. And the reason that I could talk about them so concretely is because they were peer-reviewed by scientists. And sort of the thing about science is overall, it's really built on the back of experts. So you have people that are very, very knowledgeable about a field, and they learn a lot of things, and then the people that come after them, they're building on them. And so science is really predicated on building upon what people have done in the past, as well as the rigor of not only the scientific method, which we're going to talk about in a second, but also the rigor of your peers, because you have to really trust your peers, trust their knowledge, trust the fact that they're giving something a good look over to make sure it's actually accurate. And the beautiful thing about science is when science is done right, it weeds out the bad. When science is done wrong, it promotes the bad. And so there's always a double-edged sword to science. It's really, really important. Everything you can physically do and see in your everyday life is because of science. But there's also a lot of really, really crappy stuff out there because of science. And sort of the reality of this sort of how science is done lecture that I give is every year it becomes more and more important that I sort of give it. And that's sort of an unfortunate thing. Historically speaking, we think back to World War II or something like that, where scientists were heavily trusted and doctors were heavily trusted. But now it's sort of in the age of the internet, sort of giving this lecture becomes more important because there's so much false information out there, so much false narratives out there, that really this lecture becomes one of my more important ones just because of how important it is to understand science. Because again, it's pervasive on everything that you do. You sitting here listening to me is all predicated on science done to invent the internet, right? All science, all the time. But when we think about science, as a whole, science is a collaborative effort. As I mentioned, it builds upon the backs of people that came before you, and science rarely is, if ever, is done in solitary. So this idea of the mad scientist, whether it's the doctor from Back to the Future, Rick, or Professor Farnsworth from Futurama, it's not done in solitary like this. It's not a mad scientist. It's a group of people, knowledgeable people, doctors, PhD scientists like myself that are doing this work, collaborating with one another, and trying to answer these sort of big questions. Now, sort of the basic framework we have here for how the scientific method is sort of put forth, how scientists go about approaching a problem, is really starts at the basic idea of getting a question. What do I want to answer? Now, this question could be anything. The question could be about behavior. It can be about molecular biology. It could be about cells. It could be about evolution. It could be about anything. But you have to have a question. There has to be a problem out there for you to solve, right? And so once you do that, once you identify a problem, you form a hypothesis. And this is just a well-stated question. I hypothesize that fertilizer will make plants grow better because of X. That's a hypothesis. Once you establish your hypothesis, you set up some sort of experimental design to test your hypothesis. Once you get the results of your experiment, you draw conclusions, and you do lots of math along the way. Science is linked to math super duper strongly. And either you support your hypothesis or you're not supported and you go back to the drawing board, or if it's supported, you report your results to others. Now, just to sort of show you in the best way that I physically can, I'm going to use some memes to show you how the scientific method is done, because I like to make sure people are paying attention. So we make an observation, we ask some sort of question, we form some sort of hypothesis, and that hypothesis leads us to make some sort of prediction about the system. We do some sort of experiments on this topic, and one of two things. We get results, which leads us to do one of two things. We either get, as we said, we reject our hypothesis, we go back and reform a hypothesis and repeat this whole thing over and over again, or we develop some sort of theory, we publish a paper, we give a scientific talk, and then we go back to, and this rinses and repeats itself. And science is sort of this sort of beautiful self-feeding thing, is even if you get results, you want more results. And your results actually ask more questions at the end of the day. So, scientists are never short on new questions. And if you ever wanted to talk to a scientist about their work, they would talk your ear off for a very, very long time. Because, as I mentioned, this sort of cycle is self fulfilling. You learn things, you want to learn more things. You always want to build upon what you've learned, and you also want to build upon what your peers have learned. And so to put this in, again, in a real world perspective, we have some sort of observation here. We have a plant that's not growing very well. We want to ask this question: what type of fertilizer works best? And so we grow plants with fertilizer. We grow plants with 3 different fertilizers, and we make the hypothesis that plants with fertilizer A will grow the fastest. So we plant a bunch of plants with fertilizer A, fertilizer B, and fertilizer C. We get our results, and then we come to the conclusion that, hey, fertilizer A was the best fertilizer. And now the question we can ask after that is, why was fertilizer A the best fertilizer? And then we can go about trying to answer that question. So that's sort of the basics of the scientific method. Again, it is a self-fulfilling thing. It begs you to ask more and more questions, because ultimately science is a question-driven field. Now, once you have all these results, science is primarily disseminated in the form of research articles. And again, these research articles are peer-reviewed. So anytime you see something in a journal, like for instance, this is in the journal Science. This was peer-reviewed by some really, really smart people that know a lot about the topic of this paper. But this is the primary way how science is disseminated to the public, to other scientists. There are other ways, including conferences and stuff like that, but this is the primary mechanism by which science is disseminated. And now, when you guys were doing your papers, I asked you to pick up a news article from a periodical that essentially picked up on a published journal article and write about it. And so the sort of the way that the public gets access to these things, again, is from a journal picking up a published paper like this, which again is peer-reviewed, and then the sort of the publication does their best to disseminate that information to the public. And so when we're thinking about scientific articles as a whole, again, they are peer-reviewed, and so these are something that either presents the results of an experiment or an observation by scientists. And again, it's peer-reviewed, meaning 2 or more experts in the field review the article for its rigor, for the math, the experimental design, the conclusions, the tests done, all this stuff. Someone that knows what they're doing reviews this. So it's not just sent out to, some guy working at Stop and Shop and he says, okay, that looks cool. No, it's sent out to someone, like me, that knows, knows statistics, that knows the methods, that knows experimental design, stuff like that. So this is, when you see something published, typically speaking, it is something that has undergone rigorous beating up, because sort of the one thing that scientists like to do with one another is we like to argue. And we like to argue about results. We like to argue about methods. We like to be very rigorous with one another because we hold one another to a very, very high standard. And this is sort of why sort of like big topics where there's huge scientific agreement, like climate change, are kind of a big deal because scientists, again, we don't like to agree with one another, we like to argue. But something like climate change, which has essentially unanimous support, is kind of an amazing thing. And we'll talk about climate change next week, but I wanted to just sort of put that out there because I always find it funny, you know, how much scientists like to argue and discuss things with one another. That being said, when we're thinking about how science is done and how journal articles are published, this is typically funded by the government. And so most research is government funded. So you're thinking about the National Institutes of Health, the National Science Foundation, the European and various European, Chinese, and Russian agencies use taxpayer dollars to fund science, to pay students, to pay researchers, to pay for the experimental materials, to ask these questions. And when we're thinking about sort of overall funding, it can either be government funded, as I mentioned, which is your tax dollars, or it could be private funding. And so this can come from a private organization, like, for instance, like the Bill and Melinda Gates Foundation. They're big into funding science. Google is also big into funding science. But it could also be funded by investors. Investors are paying someone to do research so they can develop a product. And so it's important to sort of think about where the funding from any scientific article is coming from, any scientific paper, any scientific talk, anything is coming from, because people can have motives, right? If you're an investor, you want to get results because you want to make money from this, right? And so there's all sorts of ways that you sort of want to evaluate where the money's coming from, because it's an important thing. If your money is funded by a special interest group, well, that might not be the best journal article published. But typically speaking, you can think of, well, if it's published in the world. And this is because journals have tiers. And so you can sort of assess the quality of a journal by what tier it's in. And you can assess this quality by an impact factor. And so if you want to know what an impact factor is, I linked the Wikipedia article here. But it basically ranks journals by how often they are cited. And the more citations, the better the journal is, the better the articles they publish, the more prestigious the journal is. So journals do have tiers. And so something like Science is at the very top of the pile. It publishes the best cutting edge research. But there's lots of other journals that publish sort of low hanging fruits, not very impactful science. It doesn't mean that they're bad, but it simply means the findings aren't as sort of robust or as important as overall. So if you even as just as a student could send an article that you wrote that was absolutely complete garbage to a predatory journal, you pay a few $100s and they'll publish it without peer reviewing it, without reviewing it. They'll just publish it. And that's what a predatory journal does. They try to make money off of publishing fake or bad science. And again, that's in contrast to a regular peer-reviewed journal that is good and you can trust. But the bottom line thinking about journals is if something is published in a journal, it doesn't mean it's perfect. You always have to interpret any scientific study with caution. You have to look at the quality of the journal. You have to look at the methods. You have to look at the statistics. You have to look at all the things that go into a scientific article to really assess its issue, to assess its quality. And I think it's important also when you're interpreting science to always put it in the context of the larger picture. So if you're viewing a scientific paper on climate change, you're reading it because you're interested, and it says climate change isn't a thing. Well, that's one paper, and then you go out and you find 5,000 other papers that talk about how climate change is happening and all the stuff about it. Well, you might want to interpret that one paper that's a naysayer against the massive library of papers that say yes with a lot of caution. So. And sort of one of the things about science as a whole is it does protect itself from error with proper experimental design and statistics. Now, when we're thinking about what proper experimental design looks like in science, there's a couple things we have to think about. So the first is being a large data set. So, and I think this one sort of speaks for itself. The more data you have, the more likely you are to get the correct answer. So think about it this way. If I flip a coin, right? If I flip it 10 times in a row and I get 10 heads in a row, is that really the probability of me? Should I really get 10 heads in a row, or does probability really say it should be about 50-50, right? 50% heads, 50% tails, right? Or if I flip a coin 100 times, well, maybe I'll get closer to that 50-50. But if I flipped a coin 1,000,000 times, probability says I should end up at that 50-50 mark. And that's what a large data set allows you to do. The more data you have, the more subjects you put in your research, the more samples you collect, the more likely you are to converge on the true answer. It allows you to be more confident with your answer. So just to put this in another sort of perspective, when they do polling, if you poll 100 people, you're less confident in your answer than if you poll 1,000 people, or if you poll 10,000 people, or if you poll 1,000,000 people. The more data you have, the more subjects you have, the more sort of points on your graph that you have, the more likely you are to converge on the true answer, and the more confident you can be in your answer. And I think the sort of the final thing I'd like to say about this data set idea, and I hope I've convinced you of this throughout the semester, is that nature is very dynamic. The molecular biology is very dynamic. So lots of samples is important for weeding through the noise that typically exists in nature. Another important aspect from experimental design is replication. And when I think about replication, I'm thinking about having multiple representatives. So if I want to answer this question of does fertilizer improve the growth of plants? Well, I want to make sure I have not just one of each type of fertilizer. I want to make sure I have dozens of each type of fertilizer so I can see the effect through multiple individuals. Because again, if I flip a coin, right, and if I get heads, well, I can't make the assumption that the coin will always come up heads. I have to flip the coin more and more and more to understand what the actual probability of flipping a coin is. And it's the same with science. If I want to get an answer from a category, if I want to know if fertilizer improves the growth of plants, I need to test this many, many, many times. And so that's what I mean through space, so doing things multiple times, but a temporal component, so doing things over an extended period of time, or doing things multiple times at different time points is important, because as again, things change over time. So it's important to capture temporal or through time variability as well. So those are the sort of the idea of collecting data. Big data replication is very, very important. The other thing that's important is having a proper control. So if taking this vaccine or this drug, right? That's what I mean by control. I need to see the actual benefits of my treatment relative to something that is not getting my treatment. And that's what we mean by control, the absence of your treatment. And then finally, the appropriate math. Science is nothing without math. And that's good and bad. I mean, math is sort of a barrier for a lot of people to understand science, but science has to have math to really understand it. Can you say something is actually mathematically different as opposed to just visually? And more importantly, are you using your math correctly? Do you have the right data set to use the test, the mathematical test you're trying to use? Or do you have enough data to draw the conclusions you're trying to draw? And math is an important thing with science. And one of the sort of important things about math that I want to discuss is this idea of correlation and causation. And this gets really tripped up when people read articles in say the New York Times or the Washington Post or wherever that are reporting on science. And science is reporting a correlation between X and Y. And just because there's a correlation between those two, that does not mean there's a causation for them. And so there's this little comic here. You know, we have this sort of guy. He says, another huge study found no evidence that cell phones cause cancer. What was the World Health Organization thinking? And guy in the sweet hat says, I think they just got it backwards. Bald guy says, huh? Well, he says, well, take a look. And he has this graph where he has cell phone users in red and total cancer incidence in black. And immediately looking at this, you should know that's not quite right. But he says, there are so many problems with that. And he says, just to be safe, until I see more data, I'm going to assume cancer causes cell phones. And that's the interpretation he's making from this. You're seeing an increase in cell phones, you're seeing an increase in cancers as well. And so just because some things increase with one another or decrease with one another does not mean they are causative towards one another. And the way you get around this idea is sampling, is how big of a sample you take and how you sample through time. So sampling through time, as well as very robust sampling size, really gets around this idea of correlation versus causation. Now, you can make anything correlated to one another. And so there's this website called Spurious Correlations. It's a really fun website to take a peek at if you have time. You could waste easily an hour on this website, but they gets at this idea where you look at correlations between two things that are not related at all. So in this case, this is one of my favorites, and this is the y-axis is the number of people who've drowned in swimming pools and the number of Nicolas Cage movies. And you can see those are pretty well correlated. They track almost identically. When there's a spike in Nicolas Cage movie, there's a spike in people that drown. Now, clearly, Nicolas Cage movies are not causing people to drown, and Nicolas Cage is probably not drowning people when he appears in movies. But there is a correlation here. There's no causative link between them, but they are correlated. And so this is sort of an important thing. Just because two things change with one another, either in a positive or negative way, does not mean that one causes the other. It means they're related, but it doesn't mean they're actually related in a meaningful scientific way. So it's an important thing to think about. A lot of people get tripped up on this idea. Just because two things are correlated does not mean that one causes the other. And that's an important thing to remember. And in newspapers and journals, like the New York Times and all that stuff, they get tripped up on this all the time. Just because there's a correlation between eating something and obesity doesn't mean one causes the other. So just keep that in mind going forward. Always be skeptical of a correlative relationship. But again, this idea of math in science is important. And it's important because you want to determine if there's a meaningful difference between two categories. And you want to assess this idea, is my difference that I'm observing between two categories due to chance? Now, we can just use a very simple example here. There's always this anecdote from teachers that students perform better on exams after lunch than they do before lunch. So you can imagine a very simple experiment where we collect data. We give a test to students before lunch, and we give a test to students after lunch, see how they perform. Well, we can calculate the average or mean here, and we can see that the mean or average here is 84% before lunch, and the mean afterwards is 85%. Now, just looking at numbers, you would say that there's definitely students are doing much better after lunch versus before lunch, but are they really? And what mathematics allows you to do is suss out whether or not there's a meaningful actual difference. Is this difference actually a meaningful difference, or is it just simply due to chance? And so that's why math is important, because visually speaking, you can say, okay, yep, there's a difference just based upon the means, but look how much variability is in this data set, right? Look how much variability there is. So can you really say that? And the statistical methods allow you to say, yeah or no, there's no difference here. And just sort of the last thing about math, one of the common things that we see in sort of scientific realm is this idea of a false positive and a false negative and a true positive and a true negative. So the idea being is if you have something that's classified as a positive and it's actually really positive, you have what's called a true positive. If you have something that's classified as a positive result, but it's actually a negative, you have actually what's called a false positive. And the same thing for negatives. You have a false negative or a true negative. So you might hear these ideas sort of kicking around, but that's just sort of want to make that note. And sort of the idea being is that, again, science is nothing without math. And again, don't get trapped up in the spurious correlations world. It's fun to look because there's all sorts of crazy things here, but don't get tripped up on it. Another sort of important consideration for science is what's called the placebo effect. And I'm sure you guys have all heard of a placebo before. It's derived from the Latin word placebo, meaning I will please. And the idea being is when you have human subjects, when they take a treatment, so say a medication, they will have either a positive negative effect because they believe they will or believe that they should. So the idea being is if I have a potential drug that treats, I don't know, my cancer or maybe nausea, right? Something less depressing. So I have a drug that treats my nausea. Well, the placebo effect states that if I will more likely than not think that the placebo, so this medication that I'm getting that has no actual medical value to it, it's just like sugar, will help my stomach, will thus make me less nauseous. And that's the idea of the placebo effect. You think something should happen, thus it does happen. You're essentially your mind willing it to happen. And that might seem like sort of a far-fetched thing, but it's a really big issue in medical science. You know, it's really, really, really difficult because people want to please doctors. They want to make sure the medication works. They think the medication should work, but in fact, it really doesn't. And so when doctors and scientists are trying to understand the placebo effect, they incorporate placebos, again, a non-medication in their study, to assess the relative effect. Because you can imagine, if I have a drug that treats nausea, well, I take the drug and it has some effect. But because of the placebo effect, if I take a placebo, again, just that sugar pill, well, it will have some effect because I think it's going to help, but how much of effect does it have relative to my actual drug? And so they include placebos in drug trials. So when they were doing these COVID-19 vaccines and medication trials and stuff like that, they always include a placebo just to see the natural effect of the placebo effect. And it can be really, really, really strong. So my mom's a hospice nurse, and she talks about all the time how she gives patients medication and they think it's going to help, and it helps them amazingly, and it really actually doesn't help, but they think it's going to help. So if you want to know a little bit more about the placebo effect, it's a kind of an interesting psychology behind it. So I linked some things here from Harvard and as well as WebMD. The WebMD one is just like hey, this is the placebo effect, and the Harvard link really talks about the actual medical effects. So that's the placebo effect. But as I mentioned, science is inherently good. The intentions of scientists are inherently good, but there is bad science out there. And the sort of the unfortunate thing about science is if you're wrong with science, it can cause people to die. It can cause people to make really poor policy decisions. It can cause all sorts of bad things, right? It's not like you make a bad decision on an investment and you lose money. Well, if you do bad science, you could potentially kill dozens or thousands of people at the same time. So getting science accurate is really, really important. And sort of the nature of being a human is there are, by and large, most scientists are good, they're ethical, and they do good science. And the people that fund science are typically good. But there's lots and lots of scientists out there that are not ethical. There are lots of people that fund science that are not ethical, and they want to make money, or they're trying to prove a point, or they're trying to do something that is not sort of conducive to all the good thing that is science. So not all science is good, not all scientists are good. And there's lots of reasons for this. There's monetary and political reasons. People are being paid to find the correct answer. This is really, really rampant in the world of the anti-vaccination movement. This is rampant in the world of climate change as well. People being paid to essentially do bad science or ignore good science to make a point. People oftentimes will suppress data. This is a big deal in the climate world as well. Cherry picking data to show the trends you want while ignoring all the other data associated with it. And this sort of some of these other ones outside of predatory journals, which we've already gone over, are things that can be like accidental, right? You can imagine that maybe I didn't have enough money to find a great study, but I still published it, right? So things like poor experimental design or use of statistics, doing something very sloppy or rushed, or even having very small sample size. These can all be things that are done accidentally, but they can also be things that are done on purpose. And ultimately, as I mentioned, this is bad, right? If you have a drug that is very, very unsafe because of any one of these issues, you could kill people. Not just kill people, you could leave people harmed for the rest of their life, right? Or if you're doing bad science and you say that this pesticide is good, well, you could poison an entire ecosystem for the rest of your life, right? So you can do some really bad things with some really bad science. And so, I've been talking about all the good things about science, but there is the bad of science. And it's not like, genetically engineering, killer orangutans to take over the world. That's not the bad science we're talking about. We're just talking about the science that is not done ethically, that's not done or funded in the right way. And so we're going to move on to the next part of the lecture now. We're going to talk about some examples of science. We're going to talk about 3 cases of bad science, 2 cases of good science. two that I think are really, really interesting to sort of think about and really, really relevant. So let's dive into the bad first. And we're going to start with the this sort of really, it's a landmark paper, but it's a landmark paper for a really bad reason. And for those of you that have ever heard of the anti-vaccination movement, this is the paper, this is the bad science that started that entire movement. So this paper, published in 1998 by Dr. Wakefield, or I should say the former Dr. Wakefield and all his colleagues in the very prestigious journal, The Lancet, looked at how the MMR vaccine, as well as gastrointestinal disease, were related to autism. And just a sort of note, here's a nice sort of article, summarizing succinctly all the bad issues that we're going to talk about in case you want to read it. And here's just sort of a history of vaccines that talks about this article as well. But what you will note is that this paper was retracted, meaning its science was assessed to be bad and wrong. Its conclusions were wrong. It was thus pulled. So it's not something we should rely on. It's bad science. So let's talk about this. So the measles, mumps, and rubella vaccine, or the MMR vaccine, is something that pretty much all children across the world get. It is one of the greatest health inventions of all time. It really, really is. People don't think of measles and mumps and rubella being bad. And the reason we don't think that is because of this vaccine. We don't see how measles ravages populations. Measles is one of the most deadly diseases of all time. And when it hits new areas, or if it hits unvaccinated people, it is very, very, very deadly. I'm going to post some slides onto Blackboard about measles so you can learn a little bit more. But just to know that this vaccine was really, really important. And it still is in modern times. Now, it was invented in the 1950s at Boston Children's Hospital. So, we do a lot of good science here, which is good. But this vaccine was used for decades without any issues. It was widely used. In the United States, it essentially eradicated measles. It was really 12 children with autism or autism spectrum. And these children had received the MMR vaccine. And so these doctors, you saw this was a whole group of them. They looked at behavior, they looked at blood, urine, and they looked at the GI tract of these 12 children. They had no controls. So they had no children that didn't receive the MMR vaccine, and they did not take any blood values from controlled children. So they just had these 12 children that either had confirmed autism or something that they expected was, they believed was autism but was not confirmed. And what they concluded from this data set is that possible environmental triggers, i.e. the vaccine, were associated with the onset of both gastrointestinal disease as well as developmental regression. And this developmental regression is autism. Now, based on what I've told you thus far, you should know right away that something is not right here. They have no control. They have 12 children, right? They don't look at things through space and time. There's a lot wrong here, right? Just on the experimental design, 12 children is not enough to draw a meaningful conclusion based upon something as important as the MMR vaccine. 12 children is not enough to draw a conclusion on anything. Not something that is important like this. Even if I was trying to understand how fertilizer affected the growth of plants, I would need way more than 12 plants to understand that effect. So just right away, this should tell you this is not okay. Something is not right here. And as we dig a little deeper, we start to learn a lot more sort of not great things about this study. And we learned later that, and this is something that was completely unknown to most of the reviewers who reviewed the paper. And so this case is peer review failed here, but the science behind it is also what is failing in this paper. Now, the children that Wakefield studied were carefully selected, and some of them were actually funded. Most of his research in this thing was actually funded by lawyers that were acting for patients who were involved in lawsuits against manufacturers of vaccines. So there's this huge scientific and monetary conflict here, right? He's trying to generate data that benefits people that are trying to sue vaccination manufacturers, which is, again, more red flags. At the time, the journal, The Lancet, said its editors actually found no evidence that the authors intended to deceive them, and they didn't sort of include those that were sort of including information how the children were selected. And it's sort of an unfortunate thing. The editors expressed this regret that aspects of the funding for parallel and related work and the existence of ongoing litigation were not disclosed to the editors. So they had all these conflicts and they were lying to the journal. And so the journal couldn't properly assess whether that was a conflict of interest here. Now, the other thing that we will learn is that Wakefield actually acted unethically and has shown callous disregard for the children. Basically, he was doing highly invasive and unnecessary tests on these children, basically, borderline torture to try to get his answer. He was essentially fishing. He was eventually fishing for answers. He had this question he was trying to answer because he was being paid to answer. Well, he was doing some little bit unethical things. And then finally, after about 6 years, as a published paper, the paper was retracted, and Dr. Wakefield actually lost his medical license for this. Nowadays, he still maintains that his study was good and stuff like that, but clearly, based upon what I've showed you, this is not good. Right? This is just not good. Right? This is not good science. There's all sorts of conflicts of interest. This is not good science. And so, Margaret Spolestra, she's a doctor. She's the director of the Ontario Autism Foundation. She says the conspiracy theory that vaccine manufacturers are hiding the truth about the MMR vaccine and autism is fueled by parents need to know what is causing autism. And this is despite the fact that no large study has ever replicated his findings. So we have this very small study that's, again, not ethically, not robust. It's showing this one thing. We do much, much larger types of science. We see the exact opposite. We see no relationship here. The Dr. Spolestra goes on to say, we know that autism has a genetic cause and that there are environmental factors that we don't yet understand, and there is enormous pressure in the field to come up with those answers. Now, I understand that's something you can empathize, right? If you have a child with autism, you want to know why. You want to know if it's something you did. You want to know if it's the environment, what happened, and that desire to know is important, right? That's what drives scientific findings. But that desire to know, that desire to get money, that desire to prove a point, this should not be sort of put forth in the science like Dr. Wakefield was doing. And just as a note, no study to date has been able to replicate his findings, even ones that are paid for by anti-vaccination groups. No one has ever been able to find a link simply because it doesn't exist. So all this robust science over time is showing that this paper, which again was retracted, was absolute crap. And if you want to sort of read up a little bit more about this, we're going to talk about vaccines next week because it's important for understanding how the COVID vaccine is going to be developed. But I do recommend, if you're interested in more about this, take a peek at this link down here from thehistoryofvaccines.org. Really good website about all things vaccine. So that's the MMR vaccine in Dr. Wakefield's group. Clearly, lots wrong here. Now, let's switch gears. Let's talk about something that is cooler, but way less dangerous to humans. And this is this idea of arsenic bacteria. Now, if you remember back from our very first week together, I gave you a problem in your homework. And I said, in scientists, they hypothesized that microbes could use arsenic instead of phosphorus in their DNA. Why is that? Now, that idea came from this paper that is bad science. And I use this because A, it's cool and I love microbes, but B, because it also gets you to think about how chemistry and biology are directly connected. Now, this study was conducted in Mono Lake in California, which is a hypersaline lake, meaning it's very, 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 very salty, so way saltier than the ocean. And the unfortunate thing about Mono Lake is it is chock full of arsenic. It has arsenic levels that if you took a sip of it, in addition to the salt killing you, the arsenic would kill you. So it's very, very toxic to life. In 2004, scientists from NASA, Arizona, and Stanford University published a paper in the very prestigious journal Science where they said, here we describe a bacteria isolated from Mono Lake that is able to substitute arsenic for phosphorus to sustain its growth. Our data show evidence for arsenate in macromolecules, proteins, DNA, that normally contain phosphate, most notably nucleic acids and protein. So the idea being is phosphorus, if you remember, is integral for the backbone of your DNA. It's integral for your proteins, and it's important for your membranes. What these authors were suggesting is that instead of using phosphorus, these bacteria could use arsenic as a substitute because arsenic was so high. Now, if you remember our discussions about the periodic table, well, arsenic and phosphorus, they sit right below arsenic sits right below phosphorus, which means that they have very, very similar properties. their electron configuration in their outer orbital is identical. And they don't weigh all that much different, and they're not that much different size. And so they're very similar. So the idea being is you could potentially replace phosphorus with arsenic because they're very similar at the atomic level. And that's kind of an exciting thing, right? Like alternate ways to make life. And this has lots of important implications. As you can see, NASA was involved for thinking about life on other planets. That if you can use arsenic, well, instead of phosphorus, well, maybe that expands the regions that you can potentially live in, or maybe there's not a lot of phosphorus and there's arsenic. And so this is the idea, and this is in the bad science category. So this should tell you a bit about why this is wrong. And this is wrong not because it doesn't make sense biologically, it's wrong just because the way the science was done and the way the peer review was done. And so the way this worked is they isolated a bacteria from Mono Lake. This lake was low in phosphorus, very high in arsenic. And what they did is they grew this bacteria in a liquid media, so it's just a liquid that has all the nutrients that the bacteria needs to grow, with arsenic and phosphorus. And they did some fancy chemistry to show that it was actually uptaken by the cell. Now, what they did after that was they extracted the DNAs and the proteins from the cells, and they showed that arsenic was present in the liquid that they extracted. Now, you should ask the question to yourself, just because arsenic is present inside the cell, does that prove that the bacteria replaces phosphorus with arsenic? And more importantly, what would you need to show what would you need to do to show that the bacteria was in fact replacing phosphorus with arsenic? Now, what you would need to show here, which they didn't show, was that the arsenic was actually incorporated into the DNA. It was actually incorporated into the proteins. All these authors were showing is that, well, the arsenic was found inside the cells, and that's it. They weren't showing it was in the DNA. They weren't showing it was in the protein. They weren't showing it was in lipids. They were just showing it was inside the cell. And you should tell by my sort of disdain for this and my negative reaction to it, because these are microbiologists like me, that this is not true. What they showed was just the arsenic was inside the cells. And that happens for any bacteria that lives in an arsenic environment. The same thing for us. If we ingest arsenic, arsenic gets inside of our cells. That's the thing. Arsenic will go inside your cells. Now, follow-up work to this showed that the bacteria could actually not substitute arsenic for phosphorus. When the scientists actually did the correct science, well, they actually did not show that the arsenic was in the DNA or the proteins. They just showed that the arsenic was inside the cell. And that's sort of an unfortunate thing. It's a really cool idea, right? It shows how linked biology and chemistry are. But if you don't do the right science, if you don't have enough data, you can't fully support your ideas. And so this is something that was just a very poor experimental design. They didn't have enough data to show what they wanted to show. And even though they had all that stuff, peer-reviewed failed them. Because a good scientist that looked at this paper would have asked the question, why didn't you show it was in the DNA? That's what good peer review would have done. And peer-reviewed in this case failed. It did not catch this very, very fundamental problem. And when scientists peer-reviewed it outside of the journal and they did more science, they showed, in fact, that it was wrong. And now, this is a bad case, but this is also an important sort of note about science, is that science doesn't try to be right all the time. Science just tries to show what is right. And if I show something and then someone comes along and proves it's wrong, that's okay. We're trying to build towards the same ideas. We're trying to understand the world. We're trying to understand nature, and we want the right answer. And getting the wrong answer is really common. But as long as we find the right answer down the line, that's what we're aiming for here. So that's our arsenic and bacteria story. And then we're going to sort of wrap our bad discussion of things by talking about COVID-19 and hydroxychloroquine. This is something that's been in the news a ton recently, especially, not just COVID, but this idea of using hydroxychloroquine to treat COVID-19. Now, hydroxychloroquine is a medication that is primarily used to treat malaria. It also has a sort of a secondary use to treat certain types of arthritis. Here's a little fact sheet about hydroxychloroquine and malaria from the CDC, if you want to take a peek at it. And so this is what hydroxychloroquine was doing. And people noticed early on that there might have been some link between hydroxychloroquine and COVID-19 recovery. or lack of death, I should say. That would be the better answer, the better thing, because recovery from COVID-19 seems to be a potentially a very, very long-term thing. But we'll talk about that next week. But it has been touted by political leaders and a very, very, very small group of scientists and doctors as an effective treatment and a potential magic bullet against COVID-19. So the question is, we need to ask is, A, is there good science behind this? And B, how did we get here? And if there is not good science behind it, where did the scientific method fail? So, and before we go on, I just want to note the FDA has conclusively said that hydroxychloroquine is not approved for treatment of COVID-19 in the United States, as says the CDC. So just want to let you know that as we know now, as far as we know now, it's not really a thing. And if you want to see, I'm sorry, I've been hitting go through slides here. I got lost. If you want to see what the FDA says about that, its press releases are down here in this link. So it started, this idea of hydroxychloroquine and COVID-19 started with this one and only study, and a whole lot of politics took over. And this is an unfortunate thing. Science is independent, right? It's trying to find answers, and politics is not that, right? Politics is trying to advance your agenda or your party's agenda, right? It's the exact opposite of science, right? Science is done blind, science is done with trying to find the answers, and politics is not that. But it started with this one article. So if you want to read the article right here, it's linked at the top. If you want to read sort of a summary here about that article, this is sort of a more layman's day thing. But this study, what they did is it claimed that those receiving hydroxychloroquine were 50% less likely to die from COVID-19, which sounds pretty amazing, right? Given that it's killed 154,000 people here in the United States as of today. That sounds amazing, right? If you could take one medication that it's decently safe, not safe in high doses, but it's decently safe. It sounds great, right? If you can get rid of this pandemic by one commonly available drug, that sounds great. It would be really great to get back to our everyday lives, right? It'd be nice for us to be sitting in a classroom together, well, you sitting and me standing in a classroom together, talking about this in person, but we're not. So what's the problem here? Why wasn't this a magic bullet? What's wrong with hydroxychloroquine and COVID-19? So to really understand this, it just starts with the experiment. That's how you identify good science. So this study that they did, they had a few hundred people. I believe it was 350 volunteers with COVID-19. And so what they did is they gave individuals either hydroxychloroquine or they didn't give them anything. So there was no placebo treatments or anything like that. So I hope that puts a red flag in your head already because of how strong the placebo effect is. So that's one. The study itself was also not randomized or blind. So the people that got hydroxychloroquine knew they were getting hydroxychloroquine. The people that didn't get hydroxychloroquine knew they weren't getting it, but other people were getting it. So that's also a big problem. And the other issue, it was not randomized. And when you do a medical study, you have to randomize things because especially as you're a physician in the hospital with people, you get to know people, you get to know the severity of the disease, and science can't be done in that way. You can't be biased to giving the sickest people or the least sickest people the medication and or vice versa and not getting the medication, right? It has to be completely random. You have to take 100 people and give half of them the drug and half not the drug, and it's all random, right? You have to do that, and you have to include that placebo effect. And so that's kind of an issue with this study. That's the biggest issue that people have, is this not random, no placebo, not blind, very, very heavily biased by the doctors picking who they wanted to give it to. And that's not a good thing. You want no bias. The other problem that this study had is those that got hydroxychloroquine, typically about 90% of the time, were getting steroids. And the people that weren't getting hydroxychloroquine were not getting steroids. So you're adding this other layer on top of hydroxychloroquine, steroids or not steroids. And for those of you that don't know how steroids work, they reduce inflammation, and they're really commonly used to treat respiratory issues. And it's a pretty common treatment that if you have COVID, you get steroids to help reduce inflammation in your lungs to make you breathe a bit better. And so you can see there's this confounding factor. If you get hydroxychloroquine, you probably got steroids. If you didn't get hydroxychloroquine, you didn't get steroids. So how can you tease apart the effect of hydroxychloroquine in the presence of steroids? You would need to add treatments to the study to say hydroxychloroquine plus steroids, hydroxychloroquine plus no steroids, no hydroxychloroquine with steroids, and no steroids, no hydroxychloroquine. That's how you'd have to do that study. Again, it would all have to be random, but they didn't do that. And that's why this study takes issue. And so, and the other thing, as I mentioned, it was a relatively small sample size. It was a few hundred people. And it was all from one area of the country. It was just in Michigan at the Henry Ford Medical Center. So it's a very, I don't know if you guys have been to Michigan and all. Michigan's all, depending on where you are in Michigan, it's primarily all just white people. It's not a diverse cohort. There's no people, real meaningful people of color in this area. There's very few women were included. So it wasn't a very diverse cohort of people. And that's a problem, right? The world as a whole is diverse. And and medicine is diverse, and the way medicine affects people is diverse. So all these things make this study wishy-washy, right? It would be really powerful if it worked, but the problem is when we do follow-up studies, and there has been about a half a dozen now. If you want to take a peek at these, there was 3 very, very large studies that were done. They showed no relationship between hydroxychloroquine and COVID. And so what this is really showing us is that some bad science led us to think hydroxychloroquine was going to work. We wasted a bunch of time investigating hydroxychloroquine when we could have been investigating other things, again, because of all bad science and even bad peer review in this case. But follow-up science to that showed that it was wrong and showed that there's likely not a big issue, no sort of relationship between the two. And this is an unfortunate thing because it's a bad study and people have died from taking hydroxychloroquine, and people have died because, the overall politicization of this sort of pandemic, but sort of an unfortunate thing. It was science that was not done right. It was picked up by the news, and it was picked up by politicians as touted as this miracle thing, when really there was not enough scientific evidence to support it. And unfortunately, what it seems like now is we'll have to wait for the vaccine next year to really curb COVID in a meaningful way, at least here in the United States. Other countries have completely eradicated it, like New Zealand. Most of Europe is doing pretty good outside of the UK. But here in the United States, because we don't social distance, because we don't do all the things recommended by scientists, we'll likely have to wait till the vaccine until next year. So it would have been really nice if hydroxychloroquine worked, but the science just doesn't support it. And no matter how much we want the science to work, we can't force the science to work. So that's why good science is important. Because you can imagine if the FDA had approved this, well, we could have had a lot more people dying from COVID. We could have tried to do crazy things like reopen even earlier and get rid of social distancing and all that stuff. That would have made this even worse. So bad science has led to people dying in this case, which is really, really unfortunate. So that's bad science. Let's talk about some good science now. And we're going to start off by talking about multivitamins. And this is good science, but it gets at the misleading. And so multivitamins are over-the-counter combinations of various vitamins, vitamin A, E, D, folic acid, blah, 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 all the different vitamins, right? And the best type of these multivitamins is obviously the Flintstone vitamins. If you haven't taken the Flintstones vitamins, they're the best. And if you're going to take a multivitamin, take Flintstones vitamins. And that's not because they're better for you or anything, it's just because they taste the best. And they're shaped like the Flintstone characters. Now, there's this societal paradigm, and this paradigm also exists in my family as well, that multivitamins give you some sort of benefit, right? Vitamins are supposed to be good for you, right? You need vitamin A, you need vitamin K, right? So taking them in the form of a vitamin tablet should be good for you, right? That's the idea. But the question is, do they actually work, and how can we figure it out? Well, science comes to the rescue here, good science. And so there's 3 big studies that we can take a look at to answer this question. And they do things that are really good, really tackling the scientific method. And so the first study was a study of 450,000 people. They found that multivitamins did not reduce the risk of heart disease or cancer. So 2 of the biggest killers here in the United States. Second, a study of 6,000 men that they tracked for 12 years found multivitamins did not reduce the risk for mental declines. Finally, a study of 1,700 heart attack survivors saw no health benefits, either future heart attacks, surgery, or even death. So we have these 3 lines of evidence. And I hope, based upon what I've told you, that these should strike you as being good. If you want to read about these, I linked this article to John Hopkins down here that sort of talks about all these that will be a little bit more informative than my just sort of telling you. But I think it's an important way to show you that here's 3 scientific studies that are very robust. 450,000 people is a huge study. 6,000 people for 12 years is a huge study, right? These are large studies over a very long period of time. And remember, good science has big sample sizes. Good science is done over time. Good science assesses wide populations of people or organisms. And these studies do it. And so what we conclude from these very, very robust and large studies, and I'll let Dr. Larry Appel from Johns Hopkins says it best. He says, pills are not a shortcut for better health and the prevention of chronic diseases. And he makes that claim based upon this data and many, many, many other studies very much like it. These are 3 very large and very prestigious studies. He further says, other nutritional recommendations have much stronger evidence of benefits: eating a healthy diet, maintaining healthy weight, and reducing the amount of saturated fats, trans fats, sodium, and sugar you eat. So what he's saying is, based upon the science that we have, these 3 studies and all this other studies like it, multivitamins are not a magic pill. They're not going to make you healthier. They're not going to make you live longer. Now, and so he's saying you can take them, but they're not going to have the health benefits you think you're going to do. Now, that being said, there are some vitamins that do have benefits, like folic acid in women. There's been tons and tons of studies that show pregnant women need folic acid to produce healthy children and all sorts of things like that. If you have a vitamin deficiency, you should certainly listen to your doctor and take multivitamins or the vitamin that targets your deficiency. So like I have a deficiency in vitamin D, so I take vitamin D to prop up my vitamin D numbers. But I do that because I have a deficiency, not because I'm taking this to be a magic pill, to make me healthier, to make me live longer. But what we know from the good science is that it doesn't really seem to work. So good science applied over very large data sets can answer questions for us. And the other sort of type of good study that we want to talk about is this idea of global obesity. Now, obesity, I'm sure you guys all know, it's being just severely overweight. It's like a BMI of, I think, 35 plus, if I'm not mistaken. And we know sort of anecdotally that this is a problem that's growing across the world, and people that are obese place a very large burden on our medical system, right? If you're obese, you're more likely to go to the hospital more often, have diseases, need more medications, and so on and so forth. So obesity is a public health problem, not just here in the United States, but globally. And so there's this anecdotal evidence that people are getting more obese over time. But how can we study? And we're going to go back to the Lancet. Again, it's a good journal, but they've published some bad stuff. But there's an article published in the Lancet a few years ago. And then here's a nice sort of article summarizing what the Lancet article talks about. Now, they took 40 years of data. So they looked at 1.5 million people across the globe. They looked at Asia, the Pacific Islands, Europe, the United States, Africa, the Middle East, Latin America, and the Caribbean. So they looked at across all continents. Well, not Antarctica, because there's like 20 people who live there, so it's not really that important. But they looked across all continents, and they looked at how the BMI of these 1.5 plus million people were changing. And so what we have here is an age standardized BMI, which there's math that goes into that, but just think about this y-axis as a BMI, and we have time. So we started in the 70s, and we go all the way up to 2014 or so. And you see that for men on the left and women on the right, in all these different regions, the trend is going up. So the BMI, not only of the globe as a whole, as you can see down here, but in Latin and Central America, Africa, the Middle East, Europe, the United States, Asia, and the Pacific Islands, the BMI is increasing over time. And so what this tells us is that the globe as a whole is getting chunkier. We're getting fatter as a society. And this is, so this is what it shows you. And you should ask yourself, why is this a good study? And you say, well, for 2 reasons. A, the sample size is huge. 1.5 million people is huge. Second, it tracks them through time. So you can look at how people change over time. We're looking at how the population changes over time. Again, that's an important. Populations are not static, they're dynamic. Traits change over time, remember. And finally, they looked across different continents. They looked across different cultures. They looked across different religions, different behaviors. They looked across all these different things, and they see the same pattern over and over and over again, increasing weight over time. So this is a really strong study. It shows increasing trends over time. It is a huge sample set. It's a really, really strong study. And so we can conclusively, from this really big study, make the conclusion that obesity rates are likely changing over time. We're likely getting more obese over time. Now, this study is not just, as I told you, don't take one study by itself, but there are other studies that show similar things with big data sets like this as well. So this study, along with others, we take them collectively to say, hey, this is a problem that's changing over time. So that's the end of our talk about good studies. As I mentioned, I picked two that I thought were interesting and very relevant for sort of average everyday things, moving away from, COVID and bacteria and stuff like that I thought, people would find broadly interesting. But let's wrap up this class by talking a little bit about conspiracy and bad science. Now, conspiracies are sort of the antithesis of science. Conspiracies are ideas about topics that have anecdotal or very little evidence. And this conspiracies, historically speaking, weren't that big a deal, right? You could have a conspiracy theory, a couple hundred people knew about it, maybe it was in the newspaper, but, it was pretty small. In the age of the internet, conspiracies surrounding science, conspiracies surrounding any number of things are super pervasive, right? You can think about, 5G towers causing COVID, vaccines containing microchips, right? Those sorts of things, right? They sound kind of far-fetched. And they are. And they're far-fetched because they don't stand with good science. Now, if there was science showing that 5G radiation caused that, that's a different story. But this is a conspiracy theory. People are seeing something. They're trying to explain it. And they're coming up with this theory. And that's just not good science, right? Good science, as we've talked about, is done with experiments. It's done with controls. It's done with sample sizes. Conspiracy theories are the exact opposite. And the thing about conspiracy theories is they're everywhere. They're pervasive across cultures, religions, nations. They are everywhere. And they touch almost everything. They touch politics, they touch science, they touch everything. But I think where they become really, really problematic is in the area of science, because conspiracy theories, if they get in the way of good science, can prevent, they can cause people to die, right? They can cause the proliferation of bad science. So when we're thinking about conspiracy theories, they do rely on two key aspects. Pseudoscience, so science that is not correct technically or is just plain wrong or interpreted wrong. And this is what we see with those that are flat earthers. They're using pseudoscience to try to prove their point. And then the other thing conspiracy theories rely on is cover-ups. And this is typically by societies, companies, or governments. And the best example of this, and one of the earliest conspiracies with cover-ups, is NASA faking the moon landing or the JFK assassination. There's all sorts of things like this. So let's talk about pseudoscience, and we'll just use flat Earth as an example. And so the idea of a flat Earth, it's not a new thing. It was the sort of prevailing idea up until about 2,500 years ago. But in 2,500 years ago, we proved, inconclusively, and we continue to prove it today, especially as we fly in rocket ships and we see how round the Earth is, that the Earth is round, and there's no ifs, ands, or buts about that. That's just the way it is. Now, people that deny the sort of spherical nature of the Earth, they say that the Earth is flat. It is surrounded by an ice on the poles. There's essentially a wall, and so the Earth is flat. And there's also other things that go along with this, like, gravity is not a thing. The Earth moves up to catch you. There's all sorts of things about this. You can go Google it if you want to know the specifics. But the basics ideas is the Earth is flat. And this whole idea is based upon the pseudosciences, what is called the Zeictic method or the Zeictic method. And the idea behind this is that visual observations are the only thing that matters. Now, the idea being is I see something. I don't see what I think I'm supposed to see. Therefore, what I see is right. So in this case, we have the skyline of Chicago, which is a wonderful city if you haven't been to it. And this is Lake Michigan. So you see the skyline of Chicago, right? Chicago is a pretty wide city. Now you would imagine if we live on a curve, why don't we see the curve of the planet, right? We're looking at a pretty wide area. Why don't we see the curve of the planet? And that's the idea of what the Zeictic method is based on. I can't see it, therefore it is not true. Now, that should sort of trip your brain a little bit from what you've learned about science, right? Because it doesn't test anything. It doesn't use math to show anything. It doesn't use experiments, right? It's just an observation. And for the people, people that are in jail for, for committing, for a crime that they didn't commit because of an eyewitness, they'll tell you that eyewitness accounts are terrible. And the reason they're terrible is because people just don't have the sort of, there's lots of reasons, but in terms of this case, you don't have the capacity to see the big picture. And when we And this becomes a problem, right? Back in the 80s, not a big deal. Back in the 1800s, not a big deal. But in the age of the internet, it's a really wide-reaching idea. There's a flat Earth society now, and it's reaching really wide because of pro athletes and rappers and celebrities and all sorts of people that you hear them say these things, and because they're famous, it carries a little weight. So, for instance, when I'm a basketball fan, when Kyrie Irving, when he was on the Celtics, he said he believed in flat Earth. I was just like, come on, bro, what's going on? And so it's a really wide-reaching idea. But again, it's something that's just visually seen. It's not based upon scientific method. They're not doing actual experiments. They're just saying, I see this, therefore it is. And as I mentioned, eyewitness evidence is not reliable. And when you actually do the experiments, when you sit down and you say, is the Earth actually flat? What would this look like? It doesn't hold up. And so this is the idea of pseudoscience. And actually, as one final note, is when people try to prove the Earth is flat, they always prove the Earth is round. All the science points to the Earth being round, flat earthers being wrong, the science they do to support it being pseudoscience. And the ultimate thing is we've been out of space. We've been into space. We have people on the moon. Every other planet in our solar system is round. And so we know this just from physical evidence, as well as the math and the science that's been done. And we've known this for 2,500 years. So again, this is pseudoscience taking a hold and driving a bunch of people to believe something that is not right. Next up is this idea of being shady with conspiracy theories. And so we'll talk about this in the context of the moon landing. And there's this whole group of people that believe that we faked the moon landing. But we do know NASA landed on the moon in 1969, and they landed on it a few other times in the subsequent years. And people assert that NASA is lying. And I think there's I think outside of, if we disregard, the pseudoscience is one thing. I don't agree with that at all. But this idea of conspiracy being that the government is lying, or a big corporation is lying, or anything is lying, it's not a far-fetched thing, right? There's tons and tons of examples of big corporations and governments lying to people to protect, one thing or another. So the idea that a big entity is lying is not that far-fetched. And you can sort of look at this, right? So Edward Snowden, right? He put forth this conspiracy theory that the NSA was spying on us, and it was proved to be correct, right? The Tuskegee syphilis experiment, and many, many others. These things that were conspiracies were then proven to be correct. So the idea of conspiracy is not that far-fetched, but the question we have to ask is, what sort of goes into this? What's the psychology? What's the science that goes into this? Now, they don't rely on science when we think about this aspect of conspiracy theory. It's not something that relies on a science or any evidence. It's mostly a feeling. There is an overall psychology behind this. People want to feel right. People want to feel vindicated. People want to believe that they've stumbled onto something bigger than them. And they also want to sort of believe that they can explain something really chaotic. So after JFK was assassinated in the 60s, if I'm remembering correctly, people were putting forth all these ideas because it was such a traumatic and crazy event. They wanted some way to explain it. And a lone gunman was not the way to explain it to them. They wanted some huge thing to explain it because it was such a huge event. And so there's some really interesting psychology behind this aspect of conspiracy theories that if you're interested, I recommend you take a peek at this article here and many others like it. But the thing about this is that even if, it's all based on feelings, not based on evidence, not based on science, right? But the thing about this is, if you start to think about the number of people that is required to keep the secret of us saying not landing on the moon, covering it up, the number of people that you would need to keep that secret is astronomical. So there's a guy that has done some math. So if you want to read the actual paper, it's down here. It's a paper in the journal PLOS One. And then there's sort of, again, another, just a news article about it. But when someone sat down and did the math, they calculated that you would need hundreds of thousands of people to keep quiet about the moon landing being conspiracy, to being fake. And ultimately, it would have been something that we would have popped the lid on in less than 4 years. So for a conspiracy theory as big as the moon landing to be, or even something like Edward Snowden's, the NSA spying, it required thousands upon hundreds of thousands of people. And relying on hundreds of thousands of people or even thousands of people to keep keep a secret is tough. And so the more people that are required to keep a secret, the less likely it is to hold. And this is true for many things, like 5G towers causing, coronavirus. That would require hundreds of thousands, if not millions of people keeping silent for that conspiracy theory to be held onto. And so there is some really interesting science behind here. If this interests you, again, I recommend taking a peek behind this. But again, this idea of being shady. It's not outlandish, but it's in the absence of good scientific evidence and the lack of good evidence overall. And so just as a final note, science is based upon gathering data and what that data means in the context of the world and the context of previous data. If I collect data that shows that climate change isn't real, that data has to be collected and thought about in the context of all the other data collected about it. Because my one study doesn't mean too much in the context of a huge library of science, right? You have to put your science in the context of the bigger picture. As a whole, science is inherently unbiased. People that do science, people that fund science are not unbiased. But the basic levels of science, it's all based upon being unbiased, being trustworthy, building upon what people before you have set forth. Good science, as it has been historically, it's to be trusted. And so when I tell you that multivitamins don't have any health benefits, I show you the data for it. There's all this good science behind it. I'm not doing it because I have any motive. I'm just showing you. This is what the science does. You can keep taking your multivitamins all you want. But this is what the science says, and this is what good science is saying. But ultimately, if I present you with something scientific, this is something that should be interpreted with caution, especially if it's new data, especially if it goes against established ideas, right? If you show me something that the Earth was created by aliens, and it's in direct contrast to what we know from geology, well, I have to interpret that with caution. And then finally, science makes everything go around. It makes everything cool. It really does. Everything cool that you have, technology, medicines, phones, it's all from science. And it's all, whether or not you realize it or not, it's all from science. And, the purpose of this course and the purpose of this lecture is to show you how good science is and how pervasive it is in your everyday life. And I really want you to understand the science, how to interpret science, how to be an informed citizen. Because, if you vote for someone that doesn't believe, some widely accepted scientific policy, you should know why that person is disagreeing with you should know why the science behind that policy that you disagree with is bad or good, right? Anyways, but with that, that'll be the end of our class. Cutting it real close today, 2 minutes close. But if you guys have any questions about anything you've seen or in general, please let me know. Otherwise, I hope you guys all have a good weekend. We are entering our final week next week, which is crazy to think about. We've done 20 classes together, got 4 more, and you have an exam. We have 1 more weekly assignment, and then we're done. So I will grade your papers as soon as I can. And if you have any questions about any grading, things like that, please let me know. Otherwise, I hope you guys have a great weekend.